All right, let's talk a little more about the DNA involved in this recombination. And I went back and checked, and I believe I misspoke in an earlier one. The slide was correct. The alpha chain is the equivalent. Alpha chain of a T cell receptor is the equivalent of the light chain of an antibody, and so it only has V and J regions. It is the beta chain, which is equivalent to the heavy chain, and has the diversity region also in it. So, all right, just so we got that cleared up in case there was any problem. Yeah. Okay, so how does the cell know which bits of DNA it should loop out? Because if this process was not very, very tightly controlled, you could imagine the mayhem that this would potentially cause. Yeah, you could lose important genes and then a cell could be dead because of this recombination. So it's very important that we only recombine and only get rid of certain regions which lead to the formation of an intact antibody or T cell receptor. And so those sequences are called recombination signal sequences, RSSs. And each recombination signal sequence is going to be recognized by the recombinase, which is called RAG1 and RAG2. It's those two proteins that are going to be together, and you'll have one copy of RAG1, one copy of RAG2, and both of them look very similar to each other, but they make a heterodimer like that with the two different proteins together. And the total RSS, recombination signal sequence, consists of three different parts. So there is a heptamer at the beginning. So this is going to be on the side that is next to the actual gene that you want to make. So next to the V gene or next to the D gene, as you like. The spacer is going to be a region in the middle, and then the nonomer, which is going to be 9 nucleotides. So hepta is 7, octo is 8, nona is 9. Yeah, just like in chemistry, and just like in Greek, which is what we're stealing from here. That's going to be 9 nucleotide sequence, and that's going to be on the far side. So farther away from the gene that we're talking about. And so at each join, we're going to have a heptamer, spacer, and nonomer in that order, with the heptamer being closest to the gene. And these can be forward or backwards. So you'll have heptamer, spacer, and nonomer on the front and the back of each little thing that needs to be joined together to make a functional immunoglobulin or T cell receptor. Now the spacers can be different. There are two different lengths of spacers. One is 12 nucleotides long, the other is 23 nucleotides long. And we have this thing called the 12-23 rule. And it just so happens that when we're going to join two regions together, we can only join a 12 to a 23 and a 23 to a 12. You can't join a 12 to a 12 or a 23 to a 23. And so this also somewhat limits the possibilities of which genes can be combined together, and just basically helps the recombinases to keep track of what they're joining together and what they should be joining together, so that they can only join things together properly in a way that leads to an authentic antibody. And so these can actually be set up either way. So in the lambda, which is one of the light chains for an antibody, you'll have the 23 here on the end of the V lambda, and you'll have the 12 on the beginning, which is what we call the 5' end of the J lambda. And so 12 and 23, we're going to join those together. And you can see how these are structured. So they've just written the 23 in there. But when you see the 23, that's the spacer. And you know that next to the gene is going to be the heptamer, so the 7 nucleotides next to the gene. And the nonomer, those 9 nucleotides, are going to be a little bit farther from the gene, so on the pointy end of the triangle. And likewise over here, you're going to have the heptamer close to the gene, the 12 nucleotide spacer, and the nonomer far from the gene. So nonomers are away and heptamers are here, right close to the gene. Now the kappa chains are actually organized in reverse. So you're still going to have 12s and 23s, and they're still going to join together in the normal way. But the 12 is on the end of the V region, and the 23 is on the end of the J region. And in the heavy chain, you've got one further permutation. You've got a 23 at the beginning, and you've got a 23 at the J region. So this means you can't just join V to J on a heavy chain, because you can't join 23 to 23. You got to join 23 to 12. And so here's our little diversity region, and that's how it gets put in there, because it's got a 12 on either end. And so this is basically like hooking up different adapters if you're trying to get your, HDMI from the computer into the HDMI input of your Blu-ray player or something like that. You need a certain set of cables, and yeah, it's basically the same way with the DNA. This is how the recombinases are able to keep track of what they're doing and do it properly. Now, there's two ways of actually doing the recombination depending on what is being joined and what direction those genes are facing in, because we can join a 12 to a 23 that's facing in the same direction, or we can join a 12 to a 23 that's facing in the other direction on the DNA, because your DNA's got genes going in both directions. And it seems like, wow, we ought to have, all of our genes for this particular process facing the same way, but we don't. And so as a result, thank goodness, the recombinase has a way to deal with this. So what it's going to do is line up a 12 and a 23 right over here, so they're right next to each other. And if they're facing opposite directions, then they'll be side by side with a little loop up here. And if they're facing the same direction, then we're just going to kind of loop out this little bit right over here. And yeah, from that point, it's actually fairly easy. We're going to join together the sequences, and yeah. In the one case, we're actually going to loop out and get rid of all the extra DNA in between these things. And so it's going to be lost from the chromosome. In the other case, we're actually going to retain those genes. They will still be there upstream of our recombination because we didn't have to loop anything out and get rid of it. So those are the two ways that can happen in this recombination. Either way, you're going to end up with a leader, a variable, and a join region stuck together. The variable and join will be just contiguous. They will run one into the other, and the leader will be right next to those. And this process is called VDJ recombination or VJ recombination. Yeah, depending on whether we're talking about a light chain or a heavy chain or an alpha or a beta. Alpha being light and beta being heavy. Equivalent. Yeah, that's the process. And so there are a couple of joints involved in here. So like right between the D and the J region, we're actually going to have a signal, rather a coding joint. And so the coding joint is going to be over here. There we go. There's our coding joint. And so this is like where the D runs right into the J, and we're actually, we need to read straight through from the D into the J with no mistakes, no weird stuff. The signal joint is going to be at the other end, where we're just like the front end of the V, for example, or the back end of the J, where it's not really going into anything. So that's going to be a, yeah, called a signal joint, non-coding. Extra chromosomal DNA is what we call that little loop of DNA that is taken out of the chromosome. And so it's like in the nucleus, it's right there beside the chromosome, but it is going to be lost and eventually recycled from the cell. And the reason is for DNA to be retained in the cell, it has to have a special signal called an origin of replication. That's how we're able to copy this thing. And if there is no origin of replication, which there's not on these little tiny loops of DNA, then they cannot be copied and they will eventually be lost. Okay, so rearrangement by inversion is, yeah, just rearrangement of gene segments during VDJ recombination. This is the one that we showed as like the second case where you end up retaining all the genes. Yeah, because they happen to be organized in a way that allowed you to do this. Junctional diversity is a neat process. So in addition to making these joins between like V and D and D and J, the recombinases that are involved will actually sometimes do their job in such a way that it leads to extra nucleotides being put in or extra nucleotides being taken out at the join site. And this is another way to get a little bit of free diversity out of a limited set of genes. And so it may well be that you have to lose or gain a couple of nucleotides at a join site for there to be, you know, in order to make the best possible antibody to a particular antigen. And so this gives us a way to potentially find that. So this is what I mean. In addition to all the genes, all those 60 million just, you know, straight vanilla combinations, We're also doing junctional diversity, so we're randomly adding and subtracting occasionally at each of the join sites. And also there's this somatic cell hypermutation, where we're just going to go in and physically mutate those genes even further. And all of this leads to, just a nearly unlimited repertoire of potential things that your antibodies can recognize. And that's good. That means that if there is an antigen out there, somehow, somewhere, some way, we ought to be able to make something that binds it. It's really just a question of, can you make it quickly enough to do some good and prevent the host from dying? And that's the tricky part. Because having an unlimited set of diversity is great, but being able to find the antibody that would actually solve a particular problem by a random process is really tricky. And the immune system essentially tries to brute force solve this problem. It just has a lot of cells simultaneously trying different combinations and hoping that one of these things works. It's, if you were designing things, this is probably not the way you would design it. You would say, oh, let's just, flip through our antibody catalog, pick out the one that works, and make a ton of it. But that is not how it happens. And this is partly responsible for some of the differences you see between different people and how they will respond to a disease. The immune system is just super complicated like that. You can never tell exactly what kind of antibody you're going to get out of any particular B cell, or what kind of T cell receptor you're going to get from any naive T cell. All right, so the recombinase proper, this is the enzyme that is actually going to do joining up of DNA segments in a way that actually leads to antibodies or T cell receptors. And the recombinase is made of one copy of RAG1 protein and one copy of RAG2 protein. And these are the recombinase enzymes themselves. So there are different ways of actually joining the ends together. So one of these ways, so the recombinases, the RAG proteins, are actually going to physically break the DNA. And your DNA actually gets broken a lot more than you would think. This is a fairly common thing. There are enzymes called topoisomerases that are involved in twisting and untwisting your DNA. And they routinely break your DNA and almost always join it back up properly. But just in expressing genes, you will routinely break both strands of your DNA, often in multiple places. And so the cell has to be really good at repairing double-stranded DNA breaks. This is important. And so it's got different ways of doing that. One of those ways is called non-homologous end joining, which is a form of double-stranded break repair. Non-homologous end joining involves randomly adding or subtracting nucleotides until you are able to get one end to join to the other end and basically make a bridge. Yeah, which seems like a crazy way to do it, and it is kind of crazy, but it's better than the alternative of just dying. Yeah, so this is the last second half-court shot, basically, of DNA repair. And it works more often than you'd think, but this is generally a way that is going to lead to more diversity in the gene, but is not necessarily always going to produce the result that you want. Essentially, you got about a 1 in 3 chance, a little less than that actually, that non-homologous end joining actually works out for you. The alternative to this is homology-directed repair. This is where you just read another DNA strand that is not broken and copy that and fill in whatever bases you need to fill in to make the join there. And that's a thing that your cells are also good at. And that's the one that leads to correct repair for the most part. And that's why we have 2 copies of each chromosome, so that we can do this homology-directed repair, rather than just relying on, crazy NHEJ. Okay, so the Ku protein, or the Ku protein, is a DNA repair protein that's going to be involved in this rearrangement process. And so this is going to be involved in all of this recombination with the recombinase. The process is also going to involve DNA-dependent protein kinase, and that's going to be involved in a variety of DNA repair pathways, but including the one that makes T-cell receptor genes. Artemis is going to be an endonuclease. So a nuclease cuts DNA in this case. And an endonuclease, endo means inside, as opposed to exo, which means at the outside. So an endonuclease cuts somewhere in the middle of a piece of DNA. And so, as opposed to an exonuclease that just trims away from the end of a DNA. And so this is going to be, involved in some of these gene rearrangements. DNA ligase. So ligation means sticking DNA back together. And DNA ligase number 4 is going to be the particular one that's responsible for joining up the DNA ends to make the coding join during VDJ recombination. There we go. Another one, these look like an alphabet soup. We'll see them on a picture, and it's going to make a little bit more sense, I think. XRCC4 is a protein that is going to function in non-homologous end joining. So this is the one that creates chaos. So it's going to bind to DNA ligase 4 and the Ku70 or Ku80, which is going to, all these proteins are going to gang up at double-stranded breaks and just kind of add and subtract things until they get a join between these strands. Then there's also, terminal deoxynucleotide transferase. So this is going to work at the end of a strand. So think the end of a broken strand. Deoxynucleotide, that's going to be DNA based, not RNA based. So DNA. So we're going to add DNA to the end, and transferase means we're actually, transferring that DNA to the end. So this is the one that adds extra nucleotides at those joins, like in the T cell receptor and immunoglobulin V region. So they can, they can lead to that increased diversity, the non-templated diversity. Okay, SCID is severe combined immunodeficiency. This is a super severe immune deficiency where you lack both B cells and T cells. So, and it is fatal. You can mimic this in mice, and one of the ways that this happens is just by deleting the RAG genes. And so if you can't do any recombination, or you can do RAG1 knockout, that is a type of SCID mouse. But there are people that are actually born with this condition as well, and these are the people that would have to live out their life essentially in a plastic bubble, just breathing purified air and eating autoclaved food. And it's really cool that you can actually keep people like this alive, in spite of them having no ability to form an adaptive immune system. So they won't be able to make antibodies, and they won't be able to make specific T cells. And so they just have to survive based on all of that innate immunity and all the other cells that don't have to undergo recombination, like a natural killer cell, for example. Okay. Omenn syndrome, it's another neat one. So this is going to be a severe immunodeficiency caused by defects. We're not deleting the genes, but we're going to have defects in one or the other of the RAG genes. And so in this case, we're going to get a little bit of VDJ recombination, but we're not going to have much. And so people with Omenn syndrome are going to have a very, very limited repertoire of T cells and antibodies. And so things like an antibody transfer from other healthy people would help these people quite a bit. Next little immune condition is going to be irradiation-sensitive SCID. So irradiation means that this was discovered by exposing cells, I believe, to UV light. And this is a type of SCID where you have problems in some of your DNA repair proteins. Irradiation, UV light, will actually cause breaks in the DNA, and it will cause some nucleotides to get chemically joined together, like covalently bonded, in a way that makes them unreadable. And so they have to be cut out and replaced. And so this is how this was discovered. It's just problems in your DNA repair proteins. So these are all things that are out there. And if you don't have any of these, just, thank your lucky stars that you got a working immune system. And it's really cool that you do. Okay, a couple other of these conditions. ataxia. So anything with taxia is the ability to move. Ataxia is a sort of an inability to move or a problem with moving. Telangiectasia. There we go. Telangiectasia. Okay, and so this is a defect in a thing called the ATM protein that's involved in DNA repair and also used in VDJ recombination. So you got a little picture here. These diseases are part of the way that we actually discovered this process and how it works. Each of these diseases is important in that way, but very, very difficult for anybody who actually has these. And the ATM is is short for the ataxia telangiectasia mutated. So the gene that gets mutated in this case, yeah, makes sense, right? And it's a protein kinase, so an enzyme that would normally add a phosphate to either a serine or a threonine amino acid. And it's also going to be there and recruited to double-stranded DNA breaks. So there we go. All right, so let's look at the entire process and, yeah, see if you can follow this through. So we're going to start out up here at the top. We've got our germline conformation. This is the DNA as you are born, normal cell DNA. So first thing that happens is the complex of RAG1 and 2 is going to bind on to one end. It's going to bind on to the 23 nucleotide spacer end, if I remember correctly. And then the next thing it does is it's going to bind on to a 12 nucleotide spacer end. And it'll be whichever one it happens to bump into first. So it may be the nearest one. It may be a farther one that's kind of looped around and just showed up at the right place. And so boom, now it's got these two things lined up, which is fantastic. And so for the coding joins and non-coding joins both, you're going to have Ku70 and Ku80 that are the Q proteins, and they're going to bind to the DNA ends. Yeah. Then so the cleavage is going to happen right back here. So we're actually cutting off those little signal bits right here. And then from this point, it's basically the DNA repair pathway. So the RAG genes just find the right things and they cut them and then they go away, which seems a little irresponsible. I don't know, don't you think? But you're going to have, yeah, the Ku proteins on actually both ends. The ends that you want to keep and the ends that you don't want, which are the signal joints. The ends you want are going to be the coding joints. And yeah, then Artemis and DNA protein kinase are going to lock down on top of that, hold things together. Terminal DNA transferase is going to process the DNA ends. It'll add or take away nucleotides as needed to make a join. And then DNA ligase 4 can finally come along once all these things have done their job and actually glue the ends together. And boom, you got it. Or in the case of a signal joint, we just don't bother attaching Artemis and DNA protein kinase. We are just directly going to bind this DNA ligase number 4, and we're going to join that together, and this is going to make that little loop that we then throw away. And it's fine that we're not as careful in joining up the ends of the loop, and it's really cool that there is a mechanism to tell which ends we're actually joining together, and for the cell to actually figure this out. All right. So here is RAG1 and RAG2 together. And so the blue one is going to be, or rather the purple one at the top is going to be RAG2, and the big blue one is going to be RAG1. And so there's going to be 2 RAG1s and 2 RAG2s together in this particular structure. And together they're going to make the recombinase. So yeah, RAG1 and RAG2. And there's going to be a zinc molecule involved in the center here that helps hold everything together. All right, so there's going to be a little hinge, yeah, region down here at the bottom. So that's right in here, where it's going to be a little bit flexible, right in this area. And this allows the molecule to reach over and bind to different sequences. So we got to find the 12 end, and we got to find the 23 end. And then that tells us that it's going to be a V and a J that we're going to join up. And so, yeah, we'll bring those together, and having this little flexible bit in the center allows the recombinase to actually bind two things that are just floating past each other. It makes it a little less rigid and a little more able to do its job. And so, we're actually going to use the zinc in the catalytic part of the enzyme. It is going to be the RAG1 that actually does the cleavage. RAG2 is going to be there to help RAG1 to keep its shape properly and to interact with the DNA, I believe. Okay. So, combinatorial diversity. All right, so this is the 60 million different antibodies. This is by combining different parts in different ways, we are able to build a wide array of different chains, basically different specificities of antibody and T cell receptor. So that's like the straight plain vanilla diversity. We're just going to combine two things and not mess anything up. Junctional diversity is when we are going to insert those non-templated nucleotides. So just random nucleotides are going to be added in there, and this is the terminal DNA transferase, you'll remember, that actually does that process. So I think that's a good place for us to pause this one, and then we'll go on in the next section. Thank you very much.